Hello, I would like to start by thanking the organization for the opportunity to present the topic Interindividual Variability in the Response to Exercise Interventions on Health-Related Markers. For the next 10 minutes, we will address the upcoming field of research of looking beyond the mean responses in exercise interventions and the importance of exercise timing. When we look at exercise prescription, in its classical approach, we use the type, intensity, duration, frequency, and progression variables to prescribe a given training to improve either physical fitness attributes or other health-related outcomes. But to improve a specific trait, there are more variables influencing the heterogeneity and variability of exercise. Some we cannot control, such as genetics, others we can partially control, exercise adherence, and one in particular we can control, but it's less explored, the exercise timing. Although exercise improves many health-related outcomes through the manipulation of the classical type and those variables, research on optimizing exercise timing is sparse. In other words, whether there is a right time of the day to trigger an optimal training response remains unclear. And that's where an understanding of the circadian rhythm comes into play. Circadian rhythms have a set of specific genes controlled by a central clock located in the suprachiasmatic nucleus on the hypothalamus, as well as other peripheral clocks in virtually every single cell on our body. All of these cells have these molecular clock genes that are comprised by a main transcription, translation, negative feedback loop involving core genes such as the clock, the BML1, the PER, CRY, and REV genes. And what do I mean by this? The day and night cycle, and mainly light, play a major role in activating BML1 and clock genes. And in turn, they will activate the CRY and REV genes, which will accumulate temporarily and form a repressor complex that will inhibit the BML1 and clock genes. This whole process is endogenous and will reset every 24 hours, or it can actually be changed depending on the day and night cycle. So why is this actually important? Because these clock genes are involved in important physiological functions such as glycemic control through the activation of the TBC1D1 and GLUT4 proteins, and on the other hand, through the SIRT and AMPK pathways, they can actually impact the stability of the PER and CRY genes and change the circadian rhythms. As you can see here on this 24-hour clock, whenever the PER2 gene peaks is where we have the lowest performance on several physical fitness attributes. So to sum it up, whenever we have the circadian rhythm and the peripheral clocks synchronized, we have an optimal and predictable functions throughout the day, whereas a desynchronized circadian rhythm due to light, sleep, feeding, or physical activity problems may lead to a plethora of chronic diseases including cancer, obesity, and also type 2 diabetes. So as an example, on glycemic control, when the circadian rhythms, healthy individuals have a high insulin-sensitive period in the morning with higher beta cell responsiveness and lower hepatic insulin extraction on this period. On average, in the morning, the glycemic values are around 5 millimoles per liter. But in contrast, when we look into type 2 diabetes, they exhibit a night to morning elevation of blood glucose with reduced insulin sensitivity across this sleep period, which is known as the dawn phenomenon. On average, for this sample, the glycemic values were around 9 millimeters per liter, which is significantly higher than healthy individuals. So the question that we actually have to ask ourselves is, and specifically for type 2 diabetes, should they exercise in the morning with a high insulin resistant state, or should they do it in the afternoon with a more insulin sensitive state? So when it comes to experimental evidence, most of the information comes from the animal model, with only 2 experimental studies addressing this issue in humans with type 2 diabetes. In this specific trial, with a crossover design, we have a sample of men with type 2 diabetes taking only metformin, and they went through a 2-week exercise protocol, either performed in the morning or during the afternoon period. So the results are quite interesting on the glycemic control, since individuals who trained in the afternoon period had the lower area under the curve when compared with those in the morning training. And moreover, and that's when it becomes quite intriguing, the morning training was in fact detrimental for the glycemic control. So this is where the research program from our laboratory comes into play to fill up these gaps in the literature, and we have a clear window of opportunity to explore the impact of exercise timing on type 2 diabetes, where so far we have only 2 studies with type 2 diabetes, only with men and on metformin, which kind of limits the generalization of these results to a wider population of people with this disease. So we're not starting from the zero. For the past 6 years, we've been working with patients with type 2 diabetes, where we implemented a 1-year randomized controlled trial in which the objective was to understand the long-term effects of different exercise intensities on health-related outcomes. In our results, we observed that moderate continuous training and the high-intensity interval training had distinct impacts depending on the outcomes assessed. But surprisingly, none of the interventions impact glycemic control, which was our primary outcome and one of the most important outcomes for type 2 diabetes. And this could actually be related with other confounding variables such as exercise timing that we've been talking about. Another thing is that most of the studies in the literature, including our intervention, usually report the average group changes, which of course are important to address and guide healthcare policies and test the efficacy of a given treatment. But even without the significant mean changes, there are in fact individuals who can still respond to the intervention. We start looking at this issue of the interindividual variability response to exercise in our own study, where we assessed whether individuals with type 2 diabetes who fail to improve cardiorespiratory fitness following a 1-year exercise intervention to improve their vascular health, either assessed through arterial stiffness and structural indices. We remade our groups, we had the control group, and then we had the high and low responders depending on if they increased their CRF above the 5% threshold. On the first glance, we can actually see a significant interindividual variability in all of the vascular health outcomes, regardless of the group. But even in those deemed low responders in CRF, we observed a favorable improvements in some important vascular health outcomes, such as the lower limb pulse wave velocity and the intima media thickness on the carotid artery when compared to the controls. However, those categorized as high responders for CRF had superior benefits in their vascular health overall in most of the outcomes. So, by looking at the data through this interindividual variability approach, we hope to have a more comprehensive understanding of the role of exercise at the individual level. To finalize, in the near future, and within the scope of our research program, we are building a new intervention study to examine the impact of exercise timing in a wider population of people with type 2 diabetes, including a bigger and more diverse sample, but also an intervention that provides a clear answer to the effects of exercise timing with different therapeutical medications, for instance, insulin, metformin, and also the medium to low term effects of glycemic control, which has never been done before. Another thing is also to actually explore and look at the group level changes, and but also the proportion of individuals that actually improve on these outcomes on the individual level. Hope you found this presentation interesting, and it helps you have a new look on exercise prescription. And lastly, I would like to acknowledge, of course, the team behind all this, my colleagues Megan, who shares this project with me, and Jill and Dinesh, doctoral students from our laboratory, and especially Professor Lishardinha, the director of our laboratory. Thank you very much.